out there and not enough places to get the vaccine, and there were issues about the capacity of the different sites in terms of being able to even store the vaccine depending on which vaccine was coming. So I presented this, and I think we have some task force with the deans that we came together, and we talked about it, and there was general support that we needed to move forward with this. So I did make more formal that we wanted to try to establish a clinic. And I mean, the current partners in El Paso are still in a limited capacity. UMC has been able to expand their ability to vaccinate, but they are a healthcare system. So that makes sense that they have people to be able to manage. But the city, the Department of Public Health, is a smaller scale operation in terms of being able to vaccinate. So I know that there are other partners like the fire department and so on, and some of the hospitals. But I felt like we could start by addressing some of the needs at UTEP. So this is where everything started. So over the holidays, we had to go through a process that was quite extensive in terms of planning. So this is an outline of what I want to cover today. I have slides for many of this, but some of the other ones, they just are going to be comments. But this is the extent of what I think are the most important pieces of what we're going to talk today. So as you could see, it's quite extensive, but I'm not going to take too long. I'm going to try to be, just present the most important information related to some of these topics. And if you have some questions, please, you can ask. I don't need to wait until the end, but I'll go through it. And if you don't have any questions, we'll have time to talk at the end, and you can basically ask questions related to any of these topics. So I'd like to start by talking a little bit about the history of vaccines. And the oldest ancient documents that we know is from the Chinese literature, where they, in China, tried to develop a vaccine for smallpox. And these were very limited information, but that's the oldest that we have. But I wanted to talk a little bit about the different types of vaccines since then. And as you could see in here, I have type of vaccine, an example of the vaccine, and some comments. So the initial vaccines were live attenuated vaccines. And an example of that is the MMR combined vaccine. And these are very effective in terms of producing an immune response. And you sometimes can get that immune response with single doses. One of the issues is that there may be more side effects associated with those. And you actually have to be very cautious if you're using them in somebody that has immune deficiency. So that's one of the questions that we ask, even for any vaccine, is in terms of the immune status of the patients. So from there are also inactivated vaccines. An example of that is the polio. And as you go down, the idea here is that you may need more booster doses. And hopefully, in some of the cases, you have less side effects. So another type of vaccine is like the type that has a subunit or recombinant type of vaccines, like hepatitis B. And then you move on, and toxoid is another example, like for diphtheria and tetanus. And finally, we have a new group of vaccines, and this is the messenger RNA, which is the vaccine that we're using in our clinic at this point. This is specifically for COVID-19. And so far, the two vaccines that are approved for emergency authorization use are a 2-dose regimen. Ideally, they have fewer side effects, And in my general opinion, that's the case. So I want to touch base on a little bit of what is the messenger RNA vaccine. But before that, I want to talk about the applications. We have to go through a number of applications to become a vaccination site. The most extensive one was with the Texas Human and Health Services. So the state require a very comprehensive document that we have to complete in order to be able to qualify as being a site where we could give the vaccine. So the qualifications emphasize what clinical personnel do we have. And they were looking for people that have training in sterile compounding, that they have vaccination training. Everybody needed to be to obtain what is called the National Provider Identifier. And there were a lot of information that had to be submitted from the qualifications of the clinical personnel. For this particular clinic, because the PrEP Act allows other people to be chief medical officers besides medical physicians or MDs, you could also qualify if you were a PharmD or a nurse practitioner. So we did not have a school of medicine at UTEP. For that reason, I was assigned as the chief medical officer for the clinic in our submission. Now, this is a temporary, PrEP. The PrEP Act is a temporary because of the pandemic. So that doesn't mean that this will be in the future the way things will be done. Another issue was quite important. It was the storage capacity for vaccine. And there were a lot of issues in terms of having the type of freezers that were necessary, ultra cold and cold, having electronic digital temperature control, backup generators, secure limited access. All these pieces were part of that application. So it took several weeks to complete that application. In order to actually get this application and get granted, we also have to submit to the NTRAC2, which is the Texas Immunization Register that is used for the state. And this is something that until now is more of a volunteer basis. Not everybody submits to this system. But for the COVID-19 vaccine, is a requirement. So you need to, within 24 hours, report all your vaccinations. And we started doing this one person at a time. It was very labor intensive. But now we were able to do this as a batch through the fixed system that the Paso del Norte Health Foundation has in place with the Health Information Exchange. So right now, every day that we do vaccinations is submitted as a batch. So it's all electronic. So that really changed in terms of the labor intense operation of submitting through NTRAC. So let me just show you a little bit about the way this vaccine comes. So we have to order through a system. It's an electronic system. It's called VAOS. And that system is for the state, and it stands for Vaccine Allocation and Ordering System. So we have to select the type of vaccine that we're going to request, the quantities, the vaccination groups that we're going to target, and so on. Now, the picture that you have there on the right side is the actual container of how the vaccine comes. So once we receive it, and by the way, we just received one yesterday and we received one today. So the container, as you could see here, is isolated with foam. It has dry ice in different places, and the vials are in trays. And this is basically what the trays look like. Each one of those trays have 195 vials. And that's the quantity that we're getting on a regular basis. That may change in the future. But once you receive this, you have to move it to wherever the storage area is, and you have to unpack it, and you have to place your vaccines in this case, it's a Pfizer vaccine. So we have to place it in the minus 80 centigrade degrees within 3 minutes of receiving it. So you have to do this very quickly. And that's part of the recommendations. Now, this storage issue may change in the future because some data has come out that maybe this is not quite necessary. But until that is official, we just have to still do it according to the official guidelines. So basically, these are the different types of fridge and freezers that we may use. For the Moderna, you could use the minus 20, but we're not receiving that. I think the state decided to send us Pfizer because we have the capacity to have the minus 80. Now, I don't know if in the future we're going to change it because it could cause a little confusion from one vaccine to the next in terms of the storage and dosing and preparation and you name it. So we do some very important inventory control, and because the cold chain, how you transport from one place to another needs to be documented, because if there is any disruption in this, you could be losing the potency of the vaccine. Now, this container does have a digital wireless electronic temperature control that tracks the movement and the temperature regulations inside the box. But once we receive it, then we have to do our own internal process of how do we move the vaccine from one place to another. So this is actually a vial of what the vaccine looks like. As you recall, back in February, or actually December of 2020, the FDA gave emergency approval use to this vaccine for people that are 16 years of age and older. Now at UTEP, because we don't have many of those individuals, and because parental consent was going to be necessary, the decision was made to only vaccinate 18 and older. But theoretically, 16 and older can get the vaccine. The clinical trials, they were multinational, and it did enroll almost 44,000 people to get the initial approval. So it was a global study where they do randomized placebo-controlled trials, and it was 2 vaccines delivered 21 days apart, and it was compared to placebo. As you could see in the back here, we do need to dilute the vaccine. We use preservative-free sodium chloride 0.9% or normal saline. And each vaccine comes with an individual vial for dilution. And this is because of quality control. Now it's frozen initially, and it comes as a suspension. So we have to let it thaw and then dilute it. So some of the results of the clinical trial showed that there was 95% efficacy at 21 days after the first dose. And the most common side effects are associated with injection site type of problems, including pain, which any IM injections will give you. Fatigue and headaches were also common, and anaphylactic reactions were very rare. And in post-approval for emergency approval authorization, the anaphylactic reactions is about 10 in a million people. So it's extremely rare. And in here, it shows you a little bit of the issues with the storage in terms of if you move it from one to another, what the life of the vaccine actually is reduced. initially it's about 5 months if you keep it frozen, but once you take it out of that environment, so you have different types of lives for the vaccine. Now, if you keep it in the shipping container, you actually can keep it there by replacing dry ice for 30 days. But for us, because we move it directly to a -80, we have about 5 months as long as it's frozen. But if you move it to the refrigerator, it's 5 days and so on. After you prepare it, it's only a few hours that you have to use it. The dose is 30 micrograms or 0.3 mLs, and it's given intramuscular. Now, just quickly, let me tell you a little bit about the virus and also how we have used this new technology to be able to produce this vaccine. So this is basically the DNA protein and the gene expression process. So usually the DNA goes through some steps and it gets to what's called the messenger RNA. And from there, it goes to the ribosome where the proteins are produced. So this same technology is that we're using is we introduce messenger RNA to produce a protein that is going to stimulate the immune system to react against the coronavirus. So the coronavirus, that we call the disease is COVID-19, actually SARS-CoV-2. And the way it works, this particular virus is that if you look at this diagram on the right side, is that the virus has a spike protein, and that attaches to a receptor on the surface of the cell. Once attaches, it can enter the cell and go through a process of using our own protein synthesis to produce more viruses, which are released back into the bloodstream. And one of the problems, as you probably know, is with the lungs. It does have an affinity to go into your lungs and cause problems, from inflammation to infection and so on. I know this is a little technical, but I think it's important that we talked about this because there is a lot of confusion about different types of vaccines. So using a synthetic process, we produce messenger RNA molecules that they're basically, the expression of what they need to do is to produce a stimulation of the immune system for that spike protein in the surface of the virus. Now we need to modify this messenger RNA to protect it from our own immune system and also to get to the right place to cause the effect that we want. So we package that using liposomal membranes and carbohydrates. And that gives stability and delivers to the right places. So it does produce high levels of the spike protein. And these are recognized for, for the immune system to respond. Now, how long this protection will last is still unknown, but we're learning more as we go forward because this vaccine was developed on a relatively short time. We don't know everything in terms of long-term. But even if we have some mutations, this technology can be adapted rapidly. It's relatively easy to produce the vaccine. The problem is to get it to the right place. So these are myths related to, that we heard out there related to this vaccine. Some people have said that embryonic stem cells are used to produce this vaccine, and this is actually not true. There are other vaccines that's the case, but not this one. Another confusion is that people keep asking is this a live or attenuated vaccine? And it's not. That's also false. Another question that people may have is that it was rushed and not properly assessed for safety. Actually, the clinical trials were extensive enough to show that this is a very safe vaccine. The questions that we still have is more about the duration of the effect, and also does it prevent transmission of the disease from one person to another. So what we are learning is that transmission appears to be another benefit. Not only controls the more severe disease, and in some cases no disease at all, but it does appears at this time that it prevents transmission to others. So these are some of the myths related to this vaccine. Now there's additional information if you want to read more about it, if you're in the technical side of things, those are some references. But let me go back to our clinic. The very first thing that we did at UTEP is to develop a questionnaire that was sent out, and the primary purposes of that questionnaire that was online and was sent through an email is basically to establish a sense of how many people at UTEP wanted to get vaccinated, and that included faculty, staff, and students. Second, we wanted to create priorities of who should get vaccinated first. And this is based on criteria that was recommended by CDC, and some of them adopted by the state in terms of high risk categories of people. So we wanted to prioritize those. The other purpose is to obtain consent to get the vaccine, and consent to also do some research for us, to develop some specific questions that we wanted to answer. And we did want to pre-screen using the CDC guidelines. And this is important because there are some people that we want to know, and I'll show you what the pre-screening is all about. Now, the importance of this is the ability for us to plan. How are we going to get the right people vaccinated, and how do we communicate to get them into the vaccination clinic? So the first thing that I want to emphasize that this is the state Texas Health and Human Services, and I did look again today if there was any changes, but this is what we have to follow. We cannot vaccinate everybody because we're limited in terms of who top priorities for vaccination are, who are the groups. So there is the 1A and also 1B. I don't know why my printer is printing. So I don't know, but that's the sound of the printer. I did not say anything to be printed there. So as you could see, we have a limited supply of vaccines, and these are the categories of people that should get vaccinated first. So these are the people that we prioritize at UTEP based on the responses from the questionnaire. Now, faculty and staff, we have a very high response rate of that questionnaire. Students, it was still 50-50. So we may have students out there that did not respond to the questionnaire that could be in these top categories, but we don't know. We cannot do anything about that. So once there is more supply of vaccines, then there will be more liberal use for other groups of people. So does anybody have any questions about this? Because this is important. Any questions? Okay, well, I'll tell you what. What we did at UTEP is that we basically vaccinated everybody that fits into the 1A and 1B that responded to the questionnaire. So after we did that, the president wanted to continue for us to receive vaccines and to continue to have a vaccination clinic. So we did reach out to other groups, including independent school districts, and asked them to do the same thing, and the same thing with the El Paso Community College. And there were a couple of other groups like alumni that, and also retirees. So we did reach out to some additional groups, but so far the primary target was UTEP, faculty, staff, and students that were in these categories. And the website, if you want to see updates, is listed in the slide there. And Elizabeth, you can share this presentation with anybody.
Excuse me.
Yeah.
If somebody was in one of these groups and was not called for the vaccination, what should we do?
There is a...
My son is a student and had a condition, and he registered through the form, but he was not called.
Yeah. So I'd recommend that you contact Sandy Vasquez, because she's the one that coordinates the actual scheduling. And we do find on occasion people that have communicated with us, and because for whatever reason, because I mean, this is electronic responses, and in terms of completing that the questionnaire, and that's all what we use. But there could be some other technical issues. But she's the one responsible for the actual scheduling.
Okay, thank you.
You're welcome. Okay, this is the pre-vaccination checklist that we follow, and it's from the CDC. So every time that there are two places that we look at this. One is from the initial emails that were sent from UTEP, people will need to complete this. And then once you come to the clinic, because things can change, we ask the participants if there's anything changed here. Several of these questions are critical. For example, if you receive the Pfizer vaccine, the first dose, you should get the second dose from the same product. And there is a difference in terms of the number of days. Pfizer 21 days apart, Moderna 28. Johnson & Johnson may receive approval as a single dose, and that's coming up because the FDA is already evaluating that data. But there are other questions. Number 3 there is important. I don't know if you could see it, but basically he asked about allergic reactions to other vaccines. And specifically, if somebody have anaphylaxis reaction and for one of the components of the vaccine, so we should not give a second dose. Or if they have a history to a previous anaphylactic reaction to a similar type of vaccine in terms of the other ingredients in it. So most of the reactions are actually not to the vaccine itself in terms of anaphylaxis, but to some of the other components in the vaccine. For example, there are other questions in here like pregnancy. Pregnancy is a high risk for a more severe disease, but there is limited data. So we cannot really recommend that people that are pregnant should get the vaccine. But what we do think is if somebody's pregnant, they should consult with their OBGYN. But because they are at high risk for a more severe disease, we can go ahead and give it to those individuals because there is no evidence of any harm to the fetus. But the data is limited. And that's, but I'm not going to go through every single question because it would take too long. But every time somebody says yes to one of those questions, we have more, we have to ask more questions. And there have to be a discussion about that. Any questions about that? Okay, so I'll keep going. Okay, this is the layout of the clinic. This is the interdisciplinary research building, the main entrance coming from campus. In here, there are some registration that needs to happen, and we do it by using a scanner, if they have the UTEP ID, that's really easy because it populates all the information once you scan the ID. We could also access that file using different ways, manually. And then they walk in through this path onto they get to the vaccination stations. So we have right now 5 vaccination stations. We are going to increase the vaccination stations. Every vaccination station does about 1 vaccine every 3 to 3.5 minutes. So you multiply that and you can see how you can get quite a few people vaccinated. Once they get the vaccine here in the stations, they move into the observation area. In here, we try to keep the distance that is necessary, 6 feet apart. We also have some education, and we do recommend everybody to get the V-Safe app, which is a CDC app that helps people track their next appointment, and also receive messages related to any concerns with side effects of the vaccine. Now that data goes to Atlanta and its national database. We also are collecting data, and we're doing it in 2 different places. During the 15 to 30-minute period that they have to be in observation, we ask them to complete a questionnaire, and that is using their cell phone. They can complete a questionnaire, and it's about the beliefs related to and practices related to vaccines in general. So we're tracking responses of that questionnaire. And this is part of the grant that we have with the College of Health Science and the College of Science related to the HPV vaccination. So this is a spin-off. Volunteers from the College of Health Science and the School of Pharmacy, and soon the School of Nursing, they play a role in different places. The vaccinators are primarily pharmacy students because they have a certificate for vaccination. We do have 2 faculty members here from pharmacy that supervise the students. We do have public health students that help monitor and do some education in this area, in the observation area. Here is our vaccine preparation area. So we do have some clinical laboratory science students and pharmacy students that help with the preparation. We do have communication needs between the vaccine stations and the vaccine preparation. The vaccine is actually stored on the 3rd floor, so we have to bring it down to this area, and we do it very carefully, documenting the transportation and the time that the vaccine is out of the freezer and so on. We track injections when they are delivered and prepared in here. That way we minimize waste and there is no misplacement. Because once you prepare the vaccine, is good for 5 hours, we actually label them with different codes to be sure that we don't leave an injection that is prepared in one of the stations and use it after 5 hours. So the label has a code that allow us to prepare vaccines in the morning, and we try to prepare them as needed. So we don't do it all as a big batch. So we start in the morning with sufficient numbers for the vaccine stations to have a flow, but then we keep preparing them. We do have an EMT person here in case there is an emergency. So far, the main concerns that we have is vasovagal reactions of people that are nervous and anxious and they hyperventilate. So we have talked to the public health students about during the observation phase to be sure that they look for the signs of people that are breathing maybe a little fast or they're anxious, and try to give them some guidance. So education and research is happening in here, and at the same time we're doing observation. We have AED in this area. We have not used it, I'm glad. So we did have a couple of hives, so they were treated with Benadryl. So that's kind of the flow. Once they're done with the observation, then they exit this way. So the vaccine preparation, there's a lot of technical things. If you take the vaccine from the -80 and you put it in the refrigerator, it takes you about 3 hours to be ready for dilution. We do have to, you can take it to directly from -80 to room temperature, and it only takes about 30 minutes to be ready for us to dilute it. But at the same time, you're cutting the life of the vaccine. So we try to minimize that. Now we used, we dress with sterile equipment, protective equipment like gown, mask, face shields, and gloves. Prepare the areas that we use with this disinfectant. They are not sterile areas, but we try to do everything to make it as sterile as possible. For the dilution, we use normal saline or sodium chloride. The vaccine is delicate, so we have to gently swirl 10 times the vaccine. You don't shake it. It does have an expiration after that of 6 hours. So this is the dose again, 30 micrograms in 0.3 mLs in a 1 ml syringe. We use 2 types of needles, 1 inch for most people. And for people that are large, we use a 1.5 inch in order to get to the muscle. And we use a 22 gauge. So it's a real thin needle. So the pain is relatively, let's say, limited. If you were using an 18 gauge, the pain will be a lot more. So we label the vaccine with name of vaccine, dose, lot number, original expiration date, and the code. And then there is that communication back and forth between stations and the vaccine preparation. So here is a student preparing, drawing the dose actually, and that's what they dress like. The face shields is primarily to protect the student from the vaccine, and in general, we want them to be protected because there is a lot of people coming and going. So that's how it's done. So the vaccine administration, actually, we have a student in here. They have a laptop. They do, again, use the barcode of the ID, scan it, and they do have the pre-screening questions that come up. They have a reminder in here in this poster of those vaccine pre-questions. In this particular case, we have a faculty member that was receiving his second vaccination. He's from chemistry. So you could see the student injecting the vaccine, and they do receive a CDC card. The first time, the card basically documents that they received the first dose, and in the back of the card is a reminder when they're going to get the second. Once they get the second, they get this little sticker that says, I got vaccinated for COVID-19. And of course, this particular faculty member knows me, so he came to say hello with the distance fist bump. So that's he offered those pictures to me. I didn't take them. So this is the vaccine observation area. It has a beautiful view, as you could see, from Juarez. So we have one of our volunteers guiding a little bit in terms of the education process and also participation on the Be Safe and our own survey. We asked them to wait 15 to 30 minutes. Now, most people are waiting 15 minutes, and that's how long it takes to do the questionnaire. So the timing is perfect to do the questionnaire. 30 minutes if somebody has a previous history of an anaphylactic reaction to other type of drugs or vaccines. So we want them to stay longer. So there is education. Part of the education is what to expect. Again, sign for Be Safe and complete the perceptions and belief research questionnaire. Now we have another research project that is more long-term, and it's going to be it's done through emails that are sent to the people that participated in the clinic, and is to evaluate effectiveness and safety long-term of this vaccine. So that's a separate study that we will be doing as a secondary analysis. Right now, I do have access to see how if we're having any particular issues. We did have one participant that received a vaccine, one dose, and soon after that tested positive for COVID-19, but it was because of an exposure. So her immune response from the first dose was not yet optimal, and of course did not have the second dose. So what we did, sorry, let me go back. What we did is basically ask them to, once they are released from quarantine, from being clear about the COVID-19 positive test, then they can come and get the second dose. So the EMT has a number of medications, and this is just to give you a sense of what they carry. They have a backpack, so they have medications for treatment of different emergency conditions for the most part. So this is an example of what medications they have in their backpack. And of course, they have other equipment and supplies if somebody needs oxygen or gauzes and so on. We did have a needle stick. And even that we're using retractable needles, which decrease the number of needle sticks. There was one case, and the EMT was able to help with that aspect. The EMT is also responsible for any serious adverse event reporting. So these are some of the reporting requirements that we have. One of them I stated already is the MTRAC 2. The other one is Texas Division of Emergency Management, and this is the total number of vaccines that were administered at the end of the day. Actually, we have to report it by 8 o'clock in the morning, the next morning. And that's how new allocations are distributed to us. If we don't complete that, we won't get any. The VAERS is a national system between the CDC and the FDA for vaccine adverse event reporting system. And there are two types of reactions, the adverse events and also the serious adverse event. And the definition of serious is that it requires medical treatment, hospital admission, and of course the ultimate worst possible reaction is if somebody dies. So far, the reactions that we have that were treated were more hives. And one of the people have idiosyncratic urticaria for so many different things, not only medications and the actual vaccine, but many other things triggers those type of reactions. We do have a handbook of operation, which is a guide, is not a policy and procedure, because there are so many things that are changing. As we go forward, there are new information that is coming out, and some of it we have to be careful that we don't try to adapt until it becomes official. But as part of that handbook of operation, it's about 50 pages long. We have some training, we have information about the cold chain, we have a specific policy about needle sticks, issues related to communications, instruction for faculty, instruction for students, etc. There's quite a comprehensive manual. And here is a team, including the faculty members are in the white coat. That's me back here. So we have 2 faculty members and a group of students that were present one of the very first days that we started the clinic. And so far we have administered more than 4,000 doses, and by the end of this week, it's going to be more than, it's almost like 7,000 doses that we're going to administer. So we're finishing the first 3 groups of second doses this weekend. So with that, I'll stop and see if there's any questions.